Hi guys, welcome back to another YouTube video. It's your tutor, Disha. Today, I'll be revising cellular respiration and ATP synthesis in an effort to boost your readiness for the upcoming CAPE Unit 2 examination. And to do so, yes, I'll be running through the following points here. I'll be talking about mitochondrial anatomy, glycolysis, oxidation of pyruvate, product fates, the Krebs cycle, oxidative phosphorylation, and lastly, I want to remind you about some key SBA exercises that you need to know. So would you believe that this small bean-shaped organelle is the powerhouse of cells? Yep, it's called the mitochondria. Now let me bring this a little bit closer to the camera so you can take a look at some of these structures. Mitochondria is double bounded. Yes, it has two membranes. It has an outer membrane and then it has an inner membrane. But I want you to look closely. The inner membrane is folded into some long protrusions. These long protruding things are called the cristae. And the inner membrane is folded into the cristae so as to increase surface area for ATP synthesis. And we're going to talk about that a little bit further down in the tutorial. What else can you see in this beautiful structure? I'm seeing some dots here. So there's some are small, some are large. So for the smaller dots, these are ribosomes. Ribosomes are involved in protein synthesis. And the larger dots, they are granules. These dots, they are suspended into a medium. This medium is called the matrix. So the matrix houses every other things here in the mitochondria. Like every other cell, the mitochondria has its own DNA. It's a circular DNA. So now it's time to talk about all of these lovely processes that constitute cellular respiration and ATP synthesis. What is the equation for respiration? It's glucose, C6H12O6, plus it's aerobic, so oxygen. This would give us products including carbon dioxide and water. The first step is glycolysis. We are going to make this sugar, this glucose, a bit more reactive. To do this, we phosphorylate the sugar, right? As the name suggests, glycolysis. We're splitting the sugar. Glycolysis, in summary, has 3 steps. The first step sees glucose being converted into fructose 1,6-bisphosphate. In the second step, this bisphosphate is split into 2 3-carbon sugars. These two are called glyceraldehyde 3-phosphate. And lastly, these 2 3-carbon sugars are oxidized to pyruvate. That's the end goal of glycolysis, to get pyruvate. For your exam, you guys are super lucky. You don't need to know the name of the enzymes. You don't need to know the names of all of the intermediates that are broken down. It is important, though, that you need to consider the currencies of these processes. For glycolysis, it uses 2 ATP molecules. Guess what? During the process of glycolysis, we produce 4. We would say that, therefore, there's a net gain of 2 ATP molecules. And it's also important to note that during the process of glycolysis, NADH molecules are also produced. Next, we need to talk about the fates of pyruvate. There can be 3 fates of pyruvate. Pyruvate can be oxidized to acetyl coenzyme A under aerobic conditions. Pyruvate can also be reduced to lactate under anaerobic conditions. And lastly, pyruvate can also be reduced to ethanol under anaerobic conditions. Let's approach the major one that we need to know for our exam: oxidation of pyruvate. Recall, we made pyruvate in glycolysis. Write the molecular formula for pyruvate: C3H3O3. This pyruvate undergoes a special type of oxidation, oxidative decarboxylation reaction. Let's break it apart, right? We hear two terms. We hear oxidation and we hear decarboxylation. It's a decarboxylation reaction because this pyruvate molecule will lose one of its carbon. Now, this 3-carbon pyruvate molecule will form a 2-carbon hydroxyethyl group. Remember now, we also said that oxidation occurred, and oxidation is the loss of electrons. So this 2-carbon hydroxyethyl group will be oxidized to an acetyl group. Those electrons lost will be accepted by the electron acceptor NAD, forming NADH. The final processing of this new 2-carbon molecule involves combining the acetyl group with a coenzyme, hence its name acetyl coenzyme A. But remember, we mentioned other product fates of pyruvate, right? So remember the other two, they were under anaerobic conditions. So let's write anaerobic. And one of the anaerobic yielded lactate. And for example, if our cells undergo anaerobic respiration in an effort to compensate energy, and that would result in an oxygen debt, and also we could produce ethanol, for example, in the fermentation process using yeast. All right, so that's it with the product fates of pyruvate. So remember, the most important one that you need to know for your exam is what happens to pyruvate in the presence of oxygen. So now we are ready to talk about the Krebs cycle. So that coenzyme A is bringing that 2-carbon acetyl sugar to the Krebs cycle. So let's make a note of that. So it's bringing the sugar, but this coenzyme A has to go back to the matrix to be combined to another acetyl sugar. Once it releases its 2-carbon acetyl sugar, it will combine with another 4-carbon molecule in the cycle called oxaloacetate to form a 6-carbon molecule. And we call this 6-carbon molecule citrate. Now, this 6-carbon citrate molecule is transformed into its isomer, isocitrate. And from chemistry, what are isomers again? They have the same molecular formula but different arrangement, right? So citrate here will go to isocitrate, which is also 6 carbons. To change citrate to isocitrate, we have two reactions. We have what is called dehydration and rehydration. Because a water molecule is being removed from a part of citrate's structure and rehydrated to another part. Isocitrate will undergo decarboxylation. Decarboxylation because this isocitrate will lose one of its carbon atom to form a 5-carbon molecule. And this is called alpha ketoglutarate. Carbon is given off as carbon dioxide. So NAD going to NADH. The same process is going to happen here to alpha ketoglutarate. This 5-carbon molecule will be transformed into a 4-carbon molecule. As usual, the carbon is given off as carbon dioxide and another molecule of NADH will be produced. Succinyl coenzyme A, NAD, NADH. And the carbon is given off as carbon dioxide. So this succinyl coenzyme A 4-carbon molecule will be transformed to succinate, which is still 4 carbons. However, as you can see, the coenzyme is removed, right? It's cleaved. Here, phosphorylation occurs. So there's the phosphorylation of GDP to GTP. Remember, this coenzyme is given off because it's cleaved. This 4-carbon succinate will be transformed into fumarate, which is also a 4-carbon sugar. Instead of using the electron carrier NADH, we're going to be using FADH. FAD going to FADH. This fumarate is transformed into malate. Finally, this malate, 4-carbon, oxaloacetate, we have NAD plus going to NADH plus H. Now, let's count our shmoney that we made. Let's start with the carbon dioxides. Or 2 carbon dioxides given off in this single cycle. Right, let's look for the NADH plus H molecules that we made. We have 1 here, and we have another one here, 2. There's another reaction here. So in total, we got 3 NADH molecules. We also made 1 FADH molecule. We add another phosphate group to GDP to form GTP. But GTP is readily converted to ATP. So we basically made 1 ATP molecule. So during this single turn of the Calvin cycle, 2 carbon dioxide molecules were given off, 3 NADH molecules were made, 1 FADH, and 1 ATP molecule. And the last process in cellular respiration and ATP synthesis is oxidative phosphorylation. Oxidative phosphorylation, as the name suggests, involves 2 processes. There is oxidation and there is phosphorylation. Oxidation, because recall from the Krebs cycle, we had made 2 lovely molecules, NADH and FADH. So they are going to be oxidized. Recall, oxidation is the loss of electrons. So they're going to lose their electrons and their protons. Their electrons get transferred to a protein complex in the inner membrane of the mitochondria called the electron transport chain. With the passage of the electrons through the electron transport chain, energy is going to be generated, and this energy is going to be used to pump the protons from the matrix into the inner membrane space. There's also going to be phosphorylation. Why? There's going to be a buildup of protons in the inner membrane space, and these protons will want to go back to the mitochondrial matrix, and to do that, they must go through another protein complex called ATP synthase. With the passage of these protons through the protein complex to the matrix, there's the phosphorylation of ADP into ATP. And this process produces a whopping 32 ATP molecules. So let's add our total ATP. So total ATP, we got 2 ATP from glycolysis. We got 2 ATP from the Krebs cycle because the cycle occurs twice. So that's 4. And we had 32 from oxidative phosphorylation. So this should give us a whopping 36 total ATPs. Then lastly, you need to revisit your key SBA exercises during the course for cellular respiration and ATP synthesis, using the respirator experiment for the germinating seeds, as well as the use of the respirometer, right? Those are 2 key SBA exercises that you need to revisit in the case of you getting them to assess in the paper 2, whether you're going to draw a graph or you're going to interpret results. That would be some good practice there to revisit the exercise.